院士好。
好。
那我们好，那因为松鹤教授，您学生我们现在找不到，他东西放在这里，那我们避免耽误时间，我们让第二位的高婉云高同学先报告。好，那高同学的指导教授是我们学校的分仪系的严瑞宏教授，也是我们学校的副校长。好，那我们请高同学先报告。
好，院士好，我是严瑞宏副校长的学生高婉云。那我今天要报告的是我博士论文的第二个题目 Molecular Effect of Lenzacumol to Reduce Angiopoietin-like Protein 3 Gene Expression and Regulate Dyslipidemia in Hepatic Cell and Ribophage。那主要是想要在肝细胞当中以及斑马鱼的系统当中去进一步的探讨从啤酒花所萃取出来的散子植化素 Lenzacumol， 它是透过什么样的分子机制抑制 NGVT-C gene 的 expression 来达到降血脂的功效。好，那心血管疾病根据 WHO 的统计，它是目前全球人死亡疾病的第1名。那每年大约会有1790万人死于心血管疾病。然而 atherosclerosis 就是周状动脉硬化，它又是主要导致心血管疾病的成因之一。那我们又称之为 atherosclerotic cardiovascular disease（ASCVD）。那在 ASCVD 的患者当中，可以很常见的看到他们的血液当中有 high level 的 LDL cholesterol and high level 的 TG。那再进一步呢，因为TG 是疏水性的，所以他们是需要 lipoprotein 携带才可以在血液当中被运送。那我们又称之为triglyceride rich 的 lipoprotein TGIL。那在血液当中代谢 TGIL 的 protein 又称之为 lipoprotein lipase LPL。那 LPL 它主要是在 monocyte 以及 adipocyte 所制造生成出来的。那LPL 它会被运送到细胞表面，然后它会结合在GPHBP1 的 protein 上面。然后这个 protein 它会将LPL translocate 到血管内腔里。那TGRL 它就会结合在 LPL 上面，那 LPL 就可以将 TGRL 代谢成 ruminant particle 以及 free fatty acid。那进一步的 free fatty acid 它就可以运送到周围的组织进行储存以及代谢。那在近期的研究中也证实说 NGVT-C 它可以抑制 LPL 的 activity。然后 NGVT-C 它是由一个叫 NGVT-L 的 family， 它是其中的一员。那 NGVT-C 它又是主要是由肝脏所生成制造出来的。那它大约是一个70 kilodalton 的 glycoprotein。那下面是它的蛋白结构式，它主要是由 SE1 的这个epitocyte 抑制 LPL 的 function。那它比较特别的是，它会在氨基酸的第225个位置上面，会经由一个furin 的蛋白所切割，那形成 N-terminal 跟 C-terminal。那同时 N-terminal NGVT-C 以及全长的 NGVT-C 都会同时抑制 LPL 的 activity。那先前研究也证实说，在 ANGPT 的 family 当中的 3,4,A 这几个蛋白，它都可以抑制 LPL 的 activity， 并且达到就是让血液当中的 TG 跟 lipoprotein 上升的现象。那在先前研究也证实说，ANGPTL 所以它可以跟 ANGPTLA 形成 complex， 并且A 可以促进3去抑制 LPL 的 activity。那此外呢，也有证实说，在 loss of function 的情况下，罹患心血管疾病的几率是降低 40% 的。那目前呢，根据 WHO 的统计，有 75% 的患者，就是心血管疾病死亡的患者，都是属于中低收入户的国家，所以我们才想说要找一个新型的小分子药物去进一步的抑制，看可不可以有降血脂的功效。那 Lenzacumol 我们的 M 是 Lenzacumol 它会透过什么样的分子机制进一步的在肝细胞当中抑制 NGVT-C 的 gene expression， 然后来达到增加 LPL 的 activity， 降低 TG 的功效。那首先呢，在结果当中，我们先证实了 Lenzacumol 在 5到40 micromolar 的浓度之间，它对于 HEMG2 细胞是没有毒性的影响。那进一步也证实了 Lensahumor 它可以抑制 ANGPTL3 的 mRNA 以及胞外还有胞内的 protein 的 expression。那此外呢，我们也进一步的验证它可以抑制 ANGPTL3 的 promoter activity， 并且它作用的区间在-250至-121之间。那在这个区间有2个gene 的2个 transcription factor binding site， 分别是 LSR alpha 以及 HNF1 alpha 这2个 transcription factor binding site。那我们也进一步的去分析它们的核蛋白，那也证实了说 Lensahumor 并不会影响这2个核蛋白的表达。所以我们就在猜测说，那是不是会影响到 LSR alpha 的 activity？ 那 LSR 它通常会跟 RSR 就是形成 complex， 然后 binding 在 LSR element 上面，然后去调控下游基因的表达。那在这边我们是分别处理了70901317以及96 retinol S 这2个 agonist， 然后且在共处理 Lensahumor 的情况下去分析对于 ANGPTL3 promoter 及 mRNA 的 expression。那发现在共处的情况下，他们确实都可以抑制ANGPTL3 的 gene expression。所以在这边我们证实了Lensahumor 可能就是透过抑制 LSR alpha、RSR alpha 的RSR 这2个 complex 的 activity 的情况下，抑制了 ANGPTL3 的 gene expression。那此外呢，我们还有去分析对于 ANGPTL4 跟 A 的这2个基因的影响，那发现他们也是可以 Lensahumor 也是可以抑制这2个 gene 的 mRNA expression。那还有分析了CETP 还有P6K3 就是 furin 这2个基因，那发现 Lensahumor 也是可以抑制这2个基因的表达。所以在 conclusion 目前是证实说 Lensahumor 它可以透过抑制 LSR alpha 以及 RSR的 activity 抑制 ANGPTL3 的 mRNA 以及胞外以及胞内的 protein expression。那在 future work 的部分，我们会进一步的验证说它是否会增加 LPL 的 activity， 然后有降 TG 的功效。那此外，我们还会就是进行用 zebrafish 的 model， 然后先喂食 normal 袋以及 high fat 袋1个月之后，然后再喂食口服喂食 Lensahumor 2个礼拜，然后再牺牲它们，然后进一步的验证 TG cholesterol 有没有影响。好，谢谢教授。
好，谢谢。
好，我们谢谢高同学的分享哈。那刘教授、刘院士，您这边要先直接给高同学一个这个指点呢，还是待会再一并呢？
我先问一下，就是因为我事实上不太不知道这个Lensahumor它是什么，对，所以要请解释一下，然后我才能想一下。
高同学刚太紧张了，来慢一点点。那是哪一个药啊
？Lensahumor 它主要是从啤酒花所存化出来的。
对，你那就它现在是已经在当药用呢，还是说是？
它目前还没有被开发成药，因为它就是一个膳食天然的植化。
那就变成为什么会做这个的原因？
为什么会做这个？因为我们在我们实验室的先前研究当中有证实说 Lensahumor 它可以有降胆固醇的功效。
这老鼠实验是老鼠的。
没有，之前也是在细胞的细
胞里面就很降低胆固醇。对 ，OK， 好。对，那就等一下再讨论。
好，那我们接下来是请对不起，刚刚那位 ，Hi， I cannot pronounce your name. How can you? So you might be talk about your name first, and then let us to know how to pronounce it.
Good afternoon, everyone. My name is Altinash, and my professor is Songho Chan professor. And today I will present my focusing PhD work, and title is The Role of the TCTP in Mouse Model Skin Carcinogenesis Induced by the Sum of Chemicals. And we shown that the statistic of non-melanoma skin cancer, and then number of the new cases, oh sorry. number of the new cases of in 2020 is significantly increasing that percentage of non-melanoma skin cancer, especially that the non-squamous skin cancer, some of cancer types is first one is the squamous skin cancer. Squamous skin cancer and basal cell carcinoma and melanoma. And squamous cell carcinoma is very common in the world, and especially induced that the UV and then some of chemicals. And then this carcinoma is located in epidermis area. We're focusing that squamous skin carcinoma, and we show that that clinical manifestation and how it looks like this is. And TCTP is our target gene. We're focusing that translationally controlled tumor protein is initially identified as a factor in cell growth and development and regulation of protein synthesis, protein degradation, autophagy, allergic and immune disorders, especially the cancer, and then promoting the K-oncogenic processes. And TCTP is highly conserved and highly expressed in all eukaryotes. And specific aim of this work is the role of the TCTP in papilloma, skin carcinogenesis induced by the chemicals, TMBA, TPA, and especially we gain the loss of function mouse model, investigate that the molecular mechanism of the TCTP in skin carcinogenesis. And we're focusing that the three specific aims. First one is the generate the transgenic mice overexpression and they characterize the phenotypes. And second aim of work is to examine the possible mechanism of TCTP involved in the regulation of the skin cancer development. And aim 3 is the pro-proliferative anti-apoptotic characteristics of TCTP, identify that the TCTP might play an important role in the skin cancer. And preliminary data has shown that this is only the control group and knockout group mice we use it, shown this data, and this mechanism is the general mechanism of how to get the knockout the mice. And this genotyping analysis checking by the PCR analysis, and first one is the knockout the group and keratin and TCTP gene we checking, and this first red line is the knockout, and second one is the hetero and the control group of the mice we checking, and then after we matching the mice of genotyping is the heterogeneous two male and female heterogeneous two mice matching, and after we get the conditional knockout and heterogeneous and control group of the mice. And the experimental setup we shown the age of the offspring is the 6 to 7 week mice using, and first shaving, and after first week is initiator of the DMBA we treating twice a week, and TPA is the promoter, and second week we started until the 14th and 21 week and 30 week three group of the mice we treated. And then unfortunately that we treating the mice 30 week group of mice is already died and 29 week. So we only shown that the 14 and 21 week treatment mice we shown, and these results shown that body weight have the body weight increasing by every week. But the control group of the mice is slightly increased, but the knockout group is not increasing, not changing, and then maybe might be this knockout group mice is highly stressed, and then they lose the weight a lot. And all that group is, and the incidence of the papilloma is also shown the 21 and 14-week treatment, and then 21 knockout the group of the percentage is very highly, and comparing that the control group. And after showing that mice dorsal skin is shown very clearly, very clean, and then after we treated 14 weeks, every time we treat the mice before we shaving the hair as well. And then now is pictures is not very clear, but the papilloma is already grown in the skin, very small size of the papilloma. And 21-week results shown that, and papilloma size is very big and then grown so fast. And we checking that first of epidermis area by H staining assay, and H staining assay, and epidermis layer is the very less than 14 weeks. Comparing that 14-week and 21-week, these results, and knockout the mice is significantly increased 21-week treatment, comparing that the 14-week treatment, and these results shown that the epidermis area of the control group is significantly reduced after the 40 and 21 weeks treatment, and knockout the group thickness was slightly increased. It is beneficial to reveal that mechanism of the TCTP involved in the papilloma skin and skin cancer. And we checking that fibrosis and the collagen by the trichia staining assay, and we also comparing that control group of the 14-week and 21-week treatment, and then there is the collagen and fibrosis is not no differences in here, and control group of the mice is the 14-week treatment is collagen is more higher than the 21-week the picture. And it's mean that long treatment will be lose the collagen and fibrosis as well. And then this preliminary data from trichia staining indicate that there are no significant differences at the fibrosis between the control and the knockout group after the short and long time treatment. And these results shown that my previous lab mate did, and postnatal first day until the 28 days, and after the knockout, mice's TCTP expression is significantly decreased without any chemical treatments. But in my results shown that with the chemicals, treatment chemicals, and then TCTP is the significantly decreased the results shown. And then why we checking that this central malpertense? Because this gene is anti-apoptotic and proliferative the central malpertense. So we checking how is the expression is. The phosphorylated ERK and beta-catenin is significantly increased in the knockout group, comparing that the treated acetaldehyde treatment, and then MCL1 is not too much differences in this 2%. So this preliminary data demonstrate that TCTP might play the role in the early papilloma development, and associated with the other central malpertense. First period of experiments, early onset of the papilloma on the knockout, and knockout the mice stayed happening during the treatment. Only survived knockout female mouse did not show big differences from the control and the knockout the group for long and short treatment. In our future work is next, we will collect the litter mates from all male 4 genotyping mice. It means control and knockout our expression rescue mice. We getting the all litter mates will be better to comparing the others results. After we collect the samples, we confirm that the sum of centromere genes and the anti-apoptotic and proliferative genes, and then also checking the JAK stat and apoptosis sum of signaling pathways in our future work. Yeah, thank you so much.
Okay, thank you. Yeah, the question is, what's the cause of the death? Because Professor Chen commented that most of the mice die, and
it is mean that we getting the sum of offspring mice that one the body weight is very small when we treating, and after we treating the dorsal skin, after they getting the too much stress, and then we also feed to them the glucose, but cannot every time, and then they will die for long time treatment. Unfortunately, 30-week treatment, we didn't get any results because they already died until the 29th week.
And the mice seem to vary quite a bit in their body weight, right? Some are much smaller than others.
Yeah, it is around the 10 gram, but my control group of mice is around 25 is average.
So is known why is that?
Yeah, we now we changing the second period of mice, mapping the strategy. We will be changed, and then let's see how is it. But my previous submate already did. She's the get the offspring is very big, around the 20. Conditions knockout is all around the 20 gram, like that. So mine is very big, and when I treated the mice, they will die so fastly, and then they stress it too much, and then scratching the skin as well. So it is our work problem, and we have to release that stress or gain the weight. But
how do you induce how is conditional knockout? What do you treat mice with to induce to make to knock out the gene?
We mating strategy, right, Professor?
Mating.
Mating strategy.
Because if you study the skin, one possibility is to just using some chemicals topically.
Yeah, chemicals we have to every week checking the body weight and
for conditional knockout.
Yeah, depends on their body weight.
Okay.
If their body weight is very less, we have to treat the less amount of the chemicals.
Okay.
Actually, I have a similar question because normally the mice around like 6 to 8 weeks old, the body weight should be around like a 20 gram. But in the beginning, your double knockout or maybe your knockout mice actually have a lower body weight than the wild type. So in this condition, maybe have the combined effect or something happen. Yeah,
I think so. So we want to changing the mating strategy again, and then see that how is the offsprings coming out the 6 to 7 week mice. If it's still very small, the mice, and we cannot treat and continue that experiment.
Yeah, so So Dr. Chen actually just as mentioned that the death, the mice dying caused by TCTP depletion thymus. So that's why I wonder, can you apply the topical? Can you do a topical application to cause deletion of TCTP only on the skin? I'm not sure it's doable. I don't have experience with that.
In our target gene TCTP, right? And TCTP is all expressed in all eukaryotes, so they're highly expressed in the plant and animal, everything. And if the TCTP knockout and how is the proceeding, we have to know that this mechanism of the work, we're focusing that. But the basic, the beginning of this work is we have a problem, so the results is not confirmed yet.
Okay.
Preliminary data is not confirmed yet. And some of publication already mentioned that, and after the knockout, without any treatment, and some of treatment chemicals and UV, as still the TCTP expression is significantly decreased. But my results is not going like that. As well that the HA staining assay is not confirmed that epidermal area is significantly decreased that for long treatment. Mine is less epidermis area than the 14-week. 21-week treatment is the less, and 14-week is the higher epidermis area. So it is the problem of maybe it is the mice or the offspring mice has a problem.
Okay, thank you.
Thank you.
Okay, so we move on to the next student. That will be Chen Zhonghui. His advisor is the Chinese medicine 的何宗荣教授。何宗荣教授也是我们慈济医院的副院长哦，也是我们中医部的这个大将。好，那我们接下来就请陈宗奎陈同学报告。我记得你也是医师吗？对，是。
刘教授好。那我的
好。
那因为我是医科所第3年的学生，那目前在慈济医院中医部的职业中。那今天要报的是我准备可能今年度要提的一个 proposal 的一个大概的一个内容这样子。好，教授，那我就开始。那这一篇最主要是要讲到说关于一个颅脑损伤的变化，经过一个中医治疗之后，一个血小板的抗原跟功能一个变化的一个观察这样子。那我们可以知道说，在2020年，卫福部指出说，在2020年其实10大死因里面，虽然我们脑损伤这部分，车祸损伤这部分不如心脏病或是癌症高，但是其实它位于第6位。那其中有46%点多左右都是因为严重的车祸所造成一个死亡的几率。所以颅脑损伤其实从轻的脑震荡到严重所谓的脑出血等等，其实都是一个非常重要的一个话题在车祸这个方面。那以目前来说，其实有分2个部分，应该是属于年轻的人。那年轻人最主要是因为他们的活动力较强，所以可能比较会有一些颅脑损伤的可能。那老年人随着年纪平均年龄、平均寿命的越来越长，其实在家里跌倒或是一些照顾方面出现一些 trouble 的话，其实也是有可能造成脑损伤。那我们自己医院其实这类的 old people 其实也是蛮大宗的一个脑损伤的一个来源这样子。那脑损伤在神经外科来说的话，其实最主要是控制它的一个脑部的 edema， 然后使它的 ICP 就是所谓的脑内的一个压力不要太高，然后在前期的监控是非常重要，因为如果在前期的监控的不好的话，其实后期会造成一个所谓的 VVB 的一个 damage 之后，会造成一个一连串的2级的一个伤害，包含严重的一个炎性的反应，以及一个所谓的脑损伤更严重的一个状态等等。这是一个脑损伤这个部分的一个解释。那 在中医的理论基础里面，其实在我们中医内经里面有指出说，理论上这类的蝶若有所最多恶血流内，大部分的这类的脑部的损伤类，其实都是跟所谓的淤血流滞在体内有关系。那淤血流滞在体内，在我们来说，我们就所谓的淤血这2个字，是我们所谓中医辩证的一个来源。那以目前来说，目前的一些研究的一些 paper 的话，例如说，因为中医在使用一些药物方面都会使用一些方剂来说，那目前常用的方剂有分为2种，一种是属于通窍活血汤，一种是属于补阳黄虎汤。那以通窍活血汤的文献指出说，其实在脑伤病人里面，如果你使用通窍活血汤的话，可以促使 NHS 跟 ADL 的一个病人状态的上升，那同时够缩短一个脑的血肿吸收的一个时间，就是它血肿时间的可以比较少一点点。那除此之外，在一些严重的脑损伤病患里面，如果我们给他吃了一些通窍活血汤，可以促进 WNT beta 的 pathway， 然后促进一些神经的 repair。所以理论上中药应该在这类病患里面有一定程度上的帮助。那另外一个方剂叫做补阳黄虎汤，那它同时能够促进一个脑内的一个血液循环之外，还可以促使大脑里面的血流量，让整体的脑的一个受损能够渐渐的减缓这样。那除此之外，它能够分泌一些神经跟促进一些神经的功能之外，可以分泌一些 CRP、编译跟 GABA 等等这些 neurotransmitter。那这些 neurotransmitter 其实跟神经的一些发育跟生长是有一定程度上的关系。那这是在中医这个方面。接下来我们要看说在血小板这方面来说的话，其实血小板其实来自于是一个 megakaryocyte， 同时它是一个没有核的一个 blood cell 这样子。那以一般现在来说的话，其实血小板被大家所熟知，最主要是在所谓凝血这个方面。可是在最近的一些文献指出说，其实它不仅仅只有在凝血方面，它其实在一些大脑神经的一些重塑啊等等，其实还有一些跟一些免疫细胞的一些互相作用，其实都扮演一个很重要的角色。所以之前的文献有提到说Fred 就是说血小板释放的所谓的那个 5-HT， 就所谓的 serotonin， 它其实可以促进一些神经的生长因子的生成之外，能够造成所谓的在大脑中的一个神经的重塑性，所以这一点对我们来说，我们觉得这是一个蛮特别的一个地方。那除此之外，这个 serotonin 也可以促使神经传导的一个速度变得更好，在一些脑伤的病患里面。那除此之外，目前也指出说，在一些神经退化性的疾病，包含了所谓的 Parkinson， 包含所谓的阿兹海默，甚至所谓的中等肌无力等等，其实都是跟血小板的一个变化其实是一个潜在性的 biomarker， 所以以我们来看，也许血小板目前来说可能是一个除了凝血机制这方面，其实应该还有扮演一个其他很重要的一个角色的一个存在。那除此之外，我们看到说在之前的研究指出说血小板的一个表现跟大脑的一个血肿的一个 value 的话，其实它有一定程度上的一个关系，所以我们觉得说血小板也许在脑损伤这类病患的一个复原方面，也许占了一个角色的一个存在这样子。好，那这是一个这张图最主要是解释说，目前血小板在一个这个应该说这个时间点的话，其实它有很多种的角色。那最上面指的是它原本的一个所谓的 clot formation， 就是所谓凝血和止血这个机制。但是中间这个所谓的 alternative 的 pathway 的话，第一个就是说，它能够释放出所谓的 PF4 这个transmitter 之后，可以促使大脑的里面的一个神经元突触的一个重塑性，这是一个蛮重大的一个发现。这样，那除此之外，下面这个话就是指说，它能够跟一些病原菌产生一些联合合作的方式，让一些外来的东西能够被抑制掉。那 PF4 也在我们这个实验中也扮演一个重要的角色，就是说我们之后想要去看说到底。我们中药处理之后，是不是 PF4 这个因子也会被一个活化的一个状态，就对了。好，那接下来其实中药有很多种，那我们希望我们找了这20年来我们中医在处方上会常用的一些活血化瘀的药物做处理。那之所以用活血化瘀的药物，最主要在于说，因为活血化瘀的药物在目前中药学的实验，现代中药学的实验指出说，第一个它能够促进血小板的一些凝血机制之外，第二个点，它能够促进一些血小板上面的一些所谓的 integrin 啊这些等等的一些细胞上的一些受体能够有活化跟一些代谢的一个机制。所以我们接着寻找到了前20个我们作用的一个药物，包含所谓的川穹、桃仁、红花、 当归，然后石菖蒲、丹参、川西跟大黄等等这些药物，想说利用这些药物的处理，看看说脑伤病人的一个状态如何，脑中的血小板的一个变化这样子。好，那这边最主要的目标在于说，我们要观察说，当我们处理在针对脑损伤的病患中处理这个我们所谓的中医药物之后，我们可以看到病人脑中的一个血小板的一个变化，这样同时也看说，在中西结合的一个治疗之下的话，我们的一个脑损伤的病人的治愈率会不会比一般非合疗的状态的话来得更好？那目前的话，实验方法的话就是如下，就是说，其实我们会收取的病患大约20到99岁的一个脑损伤的病患，但是我们取的点大概有3个点。第一个点就是说，当他入院的时候，我们会取第一个血点。那第二个点是说，当他出一般病房，就是从急重症病房出来之后，我们才取第二点。那之所以为什么会取第二点会在出病房之后才取，最主要的原因在于说，因为在脑损伤的初期，其实病人会接受比较多的西医的一个，例如说手术，例如说一些其他药物的干扰。那对于我们在介入的话，其实那个数值可能可信度不高。可是，一旦他除了一般所谓重症病房到了一般的病房的话，其实西医的目前的作为就是让他等待时间，让他脑部的一修复。那这时候如果我们处理中药的话，其实可以很明显的看到说，到底我们的中药对于病人的一个脑部的修复有没有效果。所以在出病房之后，我们再取第二个血点。那第3个点就是等到病人出院的时候，我们再取第3个点。利用这3个点的话，我们会进用一个 flow 的跑 flow 去看predictive function 的 evaluation， 包含去看说它活化的 GP2A、3A， 还有一些 surface protein 的一个差别。除此之外，还会看说一些 platelet 的 leukocyte aggregation 跟其他一些免疫细胞的一个连结状态、活化的状态是什么样子。除此之外，我会提到说，用 PL4 antibody 去看一下说整体的一个 serum NI 的一个表现是否有被活化出来。除此之外，还会看一些那个 inflammatory cytokine， 包含一些 5-HT TNF alpha、interferon gamma、IL-6跟 IL-1 等等。那除此之外，我们还会收一些medical 的 data， 然后作为一些我们佐证的一个资料。那因为目前实验IRB 刚通过，正在收案中。那以上就是我的报告，谢谢。
好，谢谢。那刚才这些中草药已经有去看过对 platelet 的影响吗？不是你的实验，我说是在过去。
之前的 data
有是吧？
对，就是多少会影响一些血小板的一些，例如说它前面受体的一些改变，或者是凝血实验的一个改变。因为本身这些都是属于比较偏活血化瘀的药，本身就会对这些凝血功能会有一些影响，这样子。
那就是 trauma 之后，当然你这个实验会看出来 ，trauma 之后 platelet 的改变也是已经有文献了，是吧？对，所以会比较新的是这些药的影响，是吧？
对，就是中药去影响之后一些改变这样子。因为在后期其实脑损伤的病患在进入一般病房的时候，其实其他的西医处置变化不大啦。就是西医的介入不会像前期他们危险性高的时候处理那么大，中后期反而他们处理其实就是比较偏休养为主。那我们想看说，那在这个期如果我们进去的话，会不会帮助它的效果能够更强这样子？
对，有一个能够想到就是这些中药不一定只是对 platelet， 对它可能有这个benefit 是以其他的原因。这是不是考虑？
对，可能就是在这些药的取用的时候，可能要更小心它其他的一些干扰性在。因为其实这些药其实大部分都是属于活血化瘀药物，但是其实它还有其他的特性存在。
对，就是说那个不太容易说排除，但是你做了才会知道，只是说到最后做完，徐立芬老师在会有问题。
是，不好意思，我问一个谢谢刘老师。我问一个问题哦 ，fundamental 问题哦，就是说这些药材的 quality 哦， 它的品质怎么去控制哦？因为有可能中药现在几乎都是大陆进口嘛，那你有没有考虑到这个 batch to batch 的困难性？哦，那怎么来 address 这个问题？还是你们医院已经有一部分的 quality control 或such 中药材的使用上面？好，谢谢。
老师好。那因为我们其实还是就是其实对病人来说，其实我们还是以我们中医的一个常规治疗为主啦。那我们使用的药还是以所谓的科学中药。那不使用水药的原因，是因为科学中药就会比较，因为有厂商的保证，那在一些量跟一些定性方面，他们会有一些保证性。那对于我们来说，到时候要回去推算或换算的时候，其实这样会比较方便。那老师说的可能是一般像坊间可能会煮水药什么，那其实误差就会很大。就像老师说，其实来源不同，其实会有一些误差。那我们尽量选的就是说，我们选固定的厂牌、固定的一个科学中药来。那对病人来说，我们定量跟定性也会比较安全。这样，以上。
OK， 所以就是完全相信那个中药那个厂商的制作的标准就对了。现在
对，因为这样副作用，因为我们 IRB 有被询问到说，其实中药处理下去会不会对病人反而有什么危险？但是因为理论上中药厂商都有 GMP 的认证，那药物方面的副作用其实也没有到很大，所以我们也比较安心给病人服用。
嗯哼。
另外建议就是，现在的趋势就是不会一次就是趋向做比较多一点，应该 high throughput，就是不 focus 在就是一两样，比如说 PF4， 我刚才你列出来那边看的不算少，就是 inflammatory cytokine 这些，对，是不是考虑可以，因为这种实验很难做，那得到的 platelet 有些可能是 confirm 过去的发现，如果说是你可以看的数据更多的话，也许一些新的可以出来，那个也许要靠一些合作就是了，但是也许不是那么难做到，就是看是不是一定是需要 fresh sample， 也许是 frozen 的或是 fixed 的，都可以做，那就是看用什么样合作方法能够去做，不是看 several 而是看 multiple 的这个，对，不一定要特定说要看什么，而是说是一种non-biased 的去看，那这目前都可能做得到，大概知道我的意思吗？
我知道，谢谢教授。因为大范围去看，也许可以看到更多东西。
对，那个的话，就是因为这是如果说慈济这边set up 没有的话，因为慈济跟中医院一直有合作，刘校长非常积极跟我们有些合作计划，那就不要客气说有什么需要的话，这边有一些就是也许有一些核心设施可以配合。不过要先知道是怎么做，那就才知道这个不太容易说是就收到的时候要运到中医院来做。如果需要做的话，那可能要 fix 这些，那先知道说怎么样是比较是要怎么 fix 才能够看这样子，对，要想一下，可以想一下。
好，谢谢教授。
因为我觉得这个基本的这个 idea 就是 trauma 之后 platelet 会是就会改变，这个 concept 很有趣说是中史就是研究两部分，一个部分是这个，一个部分是中草药的影响但是即使第一部分的话，都可能做了解的话都会有这个都是 meaningful， 对 ，OK， 好。
好，那我们下一位是周慕凡，周同学哦。那他现在人应该是在中研院还是在哪边哈？所以是直接用 online 的方式来做 presentation。那他的 advisor 一位就是刚刚有提问的徐丽芬 徐教授啊，还有就是我们慈济大学的张忠兴张教授。那我们是不是可以请我们周同学开始这个presentation？ Yeah.
Can everybody see my slide?
Yes.
Yep. Okay. I am Dongmu Fan. I am a student from the Translational Medicine, and my advisors is Professor Xu Lifeng. She is Distinguished Research Fellow of Agricultural Biotechnology Research Center in Academia Sinica. And also the other advisor is Zhang Zhongxing Professor, and she is the director of the doctoral degree program in Translational Medicine in Tzu Chi University. And today my topic is investigation of the effect of Chinese herb dendrobium species of skin cancers. And due to the patent application consideration, the genus name dendrobium species is used in here to refer to the plant currently under study. And so what is the dendrobium species? It's the traditional Chinese medicine, which 中文名字 is 石斛. And the medical use part of this plant is stem. And because most of our Chinese medicine comes from the mainland China, but this plant we have Taiwan have developed artificial large-scale cultivation in Taiwan. So the dendrobium we use in our research has the correct origin and the stable quality. And the therapeutic effect is to remove heat and replenish yin. In Chinese is yang yin qing re. And in traditional use, we call it can stimulate saliva and protect the stomach, which you can see in Chinese that 名医别录有记载它可以平胃气 and also attenuate joint discomfort and the ocular ailment. And you can see in Chinese it drives 脚膝疼痛必弱 and 明目. And this plant also has the alleviate skin issue, which can 足皮肤血热匪气, which most important part it's like the skin issue is referred to the inflammation in skin disease. And research in recently and focus on the anti-capital activity and ameliorate liver injury. And it also can suppress the inflammation from macrophage and T cell, and also has the anti-cancer ability in lung cancer, breast cancer, bladder cancer, or pancreatic cancer. But the current research stage in trying to separate the active ingredient or small molecule, as well as the preliminary biological activity. And now the non-bioactive compound, including the glycoside, bibenzyl, phenylephrine, alkaloid, and the polysaccharides. And not too many papers address on the alleviate skin issue. So recent study of the dendrobium species in type of skin disease, including the atopic dermatitis, anti-psoriasis ability, and anti-tyrosine activity. And also, I only found one article to mention about the single compound-induced apoptosis in melanoma cell. So there are few studies of dendrobium species of skin cancers, and the effect of molecular mechanism are not well understood, especially the skin cancer, including non-melanoma skin cancer, such as the basal cell carcinoma and cutaneous squamous cell carcinoma. And this cancer compromises 90% of human malignancy. And also the basal cell carcinoma and the squamous cell carcinoma are the first and the second most common malignant tumor. So inspired by the efficacy of the traditional Chinese medicine book on the treatment of the skin disease, we would like to further explore the therapeutic strategy of dendrobium in skin cancer. So we hypothesize that the dendrobium species contain bioactive small molecules that have a protective effect against the steel carcinogenesis or tumorigenesis. So our three specific, and one is using the bio-guided fractionation to confirm the potential to against skin cancer. And specific N2 is isolate the effective small molecule and confirm their chemical fingerprint and chemical identification. And the last part of the end is to elucidate the in vivo efficacy of small molecule in the animal disease model and their molecular mechanism. So it's my preliminary result. Using the bioguide fraction of the dendrobium species, we have collected the different part of this plant, and including the stem and flower part, and use the ethanol extraction to acquire the crude extract. And then we further to examine the chemical profiling using the HPLC, and then doing the MPLC for enrichment for the suppression. And then this affected suppression or enrichment to do the further chemical identification or fingerprinting. And in the meantime, the crude extract and the suppression enrichment are also to exploring their bioactivity assay, including their ability to affect on the cell proliferation or inflammatory and the cell motility, and using by the MPT assay and inhibition and adhering, and set this platform to quickly screen their ability to affect skin cancer, and let us to know how to choose the effective small molecule to do in the in vivo animal model and molecular mechanism. So the crude extract to do in the analytical HPLC profile, you can see in the figure that they have different character of the component. That the flower extract shown in red line has the more hydrophilic component than the stem extract, and showing blue line. And also we further use the MPLC subfraction to enrich the nice subfraction to do the further analysis. So about their anti-cell proliferation of the crude extract, we test this in the A cell line, including 5 of the mutant melanoma and the mutant keratinocyte, such as the A375, like the BRF mutant melanoma cell, and the drug-resistant cell like A375R acquire varroa-infected the drug-resistant A375 cell. And also we use the PDV, like the most HRAS mutated the keratinocyte. And showing the table, you can see that compared to the normal cell line, that the HACA-T, which is human keratinocyte, and the CCD960SK is the normal human fibroblast cell, and the most microphage, the row cell. Compared to this normal cell, the crude extract of the stem and the flower has more anti-cell proliferation of this tumor of this mutant cell. As you can also see, it's shown in figure that both stem and the flower extract have more anti-cell proliferation effect of these mutant keratinocyte and melanoma cell. And the same character also show in the subfraction enrichment of the stem and flower, especially the stem fraction 5 to 7 and flower subfraction 5 to 7. they also have the ability to affect more in the mutant keratinocyte and melanoma. And we choose the stem fraction 6 and the flower fraction 5 as the most effective suppression to continue the chemical identification and the chemical fingerprint. And about the cell migratory ability, the Cruactra, both stem and the flower, also show the inhibition PDV address mutant cell migratory in dose-dependent manner. So in my first year of the PhD, I'm doing a bioguided fractionation to confirm the protection against the skin cancer. And in next year, I want to isolate the affected small molecule and confirm their chemical fingerprint, and further to test the in vivo efficacy of this molecule in animal model and find out their molecule mechanism. So as being a Chinese medical doctor, I'm very proud to join the translational medicine program and to explore more Chinese medicine to treat in skin cancer. And that's my report today. Thank you.
Thank you. So in vivo, you mean topical or systemic?
Sorry, I didn't.
In vivo, you say in vivo, you're talking about the topical application or systemic, oral feeding, or 是用吃的还是
We want to test in the two-stage mouse animal model. So we try to use not oral, but using the
topical?
Why?
Oh, yes, Gavaji. Yeah.
Yeah.
So it's known. You said that few studies on this species.
Yeah.
But it's I'm not Xu Lao Shi is an expert in herbal medicine. So why do you think you are able to find active compound there if you have not tested the whole extract? Yeah, usually you pursue isolation of compound because it's known that the whole extract is effective in treating skin cancer. Literature that you mentioned that it's very few studies, but is anything there to support that there must be something active in there?
Because in tradition, I can show my. Because in traditional Chinese medicine, they have many medical books record that they have effective on the Shihu, this plant to treat the skin disease. They also mentioned it in 猪皮肤血热. And this 皮肤血热 in Chinese medicine, we think it's associated with the skin inflammatory associates disorder. So because because the tumorigenesis or the tumor progression is associated with the like the slow inflammatory effect and the inflammatory microenvironment in the body. So the traditional Chinese book is mentioned they can affect it on the skin inflammatory disease. And also recent research have focused on and have revealed that they have the compound or Cruactra to treat the lung cancer, the other cancer type. So combine this consideration, I think they have the potential to treat the skin disease.
木凡, you may want to directly answer based on your current data.
Okay.
Because in terms of crude extract to our experiences from the previous study, because this is a total crude extract.
Yeah.
It shows the IC50 value below 100 microgram per ml. So to our consideration, such activity is quite significant if we compare to other crude extract prepared from different herbal medicine. So from some theoretical or the human use experiences, I think this demonstrates that we have confidence we can identify active component from the plant extract or the dendrobium, no matter it's from stem or from flower. So your preliminary screening show the potential for it. So you got to answer directly, not reiterate the old saying or the traditional use.
Okay,
yes,
I see.
That's a very preliminary hypothesis. Yeah, let's.
Still, even I no, it's not. I'm not. I do not study herbal medicine. Although I'm very interested. Is it reasonable to, as a first step, to show in vivo activity? Even that is a big deal. I think if I see, as a dermatologist, if I see extract confirm all the literature to show it's effective in skin cancer, it alone might be very significant. It would be something to talk about. Then say what is actually active compound. Even without identifying active compound, just by the fact that you can have anti-cancer or treatment, a therapeutic effect of a crude extract. As long as it's safe, there's no toxicity. It's a important step. Yeah, but I do not know whether herbal medicine that's a traditional first step or not. Because compound may not be active compound, may not be enough. However, then I would question whether you can still hope that you will get isolate some active compound.
Yeah, I think I totally agree with what the Professor Liu just mentioned. That's why I urge Mufan to do the in vivo animal test as soon as possible.
Okay.
Yes.
Yeah.
And because the first year students just have a very fragmented time. So I plan him to start from the cell-based assay and also extract preparation and some HPLC work first. And she's going to do the in vivo animal study very soon. Yes.
Okay. Good. So,
yeah.
So
yes. So in parallel, if we can identify active component responsible for the in vivo anti-cancer effect, that would be perfect. Yes.
Okay. All right. Okay.
Yes.
Yeah. Thank you.
Thank you.
Okay, so we will move on to the next and the final student presentation. So the next presenter is the Mr. Gabriel, and his advisor is our president, Professor Yijun Liu. So just please, Gabriel, give the talk.
Thank you, distinguished faculty. Thank you, Professor, and everyone present. My name is Ayo Ibiayo, and my advisor is Professor Ingrid Liu, the esteemed president of our university. And the title of my work is Preliminary Investigation on the Effect of Dendrobium catenatum water extract on HT22 mouse hippocampal neuronal cells, and then in 3X TGAD mice. So now we already know that Alzheimer's disease is characterized by two pathological hallmarks, senile plaques, and then also neurofibrillary tangles as well. So which basically affects some regions in the brain that the temporal lobe, which has to do with the physiological functions for listening, memory, learning as well, and then also language. Then the hallmarks can be seen in the hippocampus, where we find that the fibrillar proteins of amyloid, they aggregate and then they cause shrinkage of the hippocampus. And also they have enlarged vesicles. And also on the external aspect, we have the hyperphosphorylation of the tau neofibrillary tangles. And then on the external level, and then internally the tau aggregates, and then externally we have the amyloid beta plaques as well. So also we already know that in Alzheimer's disease, it has a pleiotropic different cascades of pathological molecular mechanisms. For this, we can see that it activates microglia cells to induce neuroinflammation as well, which is part of my focus, and also autophagy dysfunction as well, which is part of my focus. So Dendrobium catenatum, I think like the last speaker just mentioned, is we are using the water extract, and it is one of the known traditional Chinese medicines that is largely found around Asia, India, Japan, and also in Europe too, and in the US. So I'll just go quickly to some of the therapeutic effects of Dendrobium catenatum. So it has been shown to attenuate atherosclerosis, and then also it plays a role in antioxidation and anti-inflammation as well. And in aging, learning, and memory, it enhances it as well. So based on these references, my aim is to investigate the effect of the water extract of Dendrobium catenatum on amyloid oligomers induced inflammatory responses and apoptosis, and autophagy in SHSY5Y neuroblastoma cells, which is a human neuroblastoma cell line, and then also HT22 mouse hippocampal cell lines. And then that's for the cellular level. For the in vivo studies as well, I want to know the role it plays on glia activation, autophagy, and then cognition using the 3XTD AD mice, transgenic mice. So for my preliminary result, first, we want to know if this water dendrobium extract has maybe cytotoxic effect at the cells, on the cells first, before we now proceed into the in vivo studies. So at the preliminary level, we did the MTT assay, and then we found out that at this dose screening, it showed no cytotoxic effect. Also, we confirmed it with the ATP assay, and then we found out that also it doesn't have any toxic effect on the maybe mitochondrial biogenesis. So next, we now discover that when we treated this compound, that is Dendrobium catenatum water extract, alongside with amyloid beta at this dose, 5 micromolar, we found out that it was able to protect the cells against amyloid beta toxicity after 48 hours treatment. So then for the in vivo study, for my future study, next now, which is currently ongoing, I want to perform immunocytochemistry, and then I want to check for NLRP3 activation and NF-kappa B, that is for the inflammatory markers, to see if DCW water extract can actually mitigate its activation. That's for my future study, which is currently ongoing. And then to in vivo, which is basically the strength of our lab. First, for the cognitive functions, which has to also do with fear memory, my first aim was to first characterize this deficit in the 3XTD AD mice to show if the mice, when exposed to contextual fear environment, maybe it's going to show difference between the wild type and the 3XTD AD mice. And then also, the next one is to check for working memory test and also spatial learning memory test as well, using the T maze and then the Morris-Water maze. The OFT test is for the locomotor activity of the mice, which we'll first use to determine if the mice has any locomotor impairment. so that we might be able to determine if it's going to confound our experiment or not. So because of the shortness of the time, I didn't include the OFT test, but it showed no difference. The mice had no locomotor impairment. So the result is not shown here, but to the contextual fear conditioning, after we subjected the mice, we exposed them in this paradigm of experiment. We normally induce, we will expose the mice to unconditioned stimulus, which is a shock for 3 intervals, and then we will train the mice with the shock and expose them to the same context. And once this is done, we will ensure that they all learn. So from this experiment, we found out that they all learned, that is the 2 group of the mice that learned. And then when we now re-expose them to the context without the conditioned stimulus, we found out that both of the mice expressed the same, almost the same freezing percentage. So there was no difference between them. So next, we then move on to the next experiment, that is to confirm for the working memory and then also for the spatial memory. So for the working memory, we conducted first the OFT test, and then we realized that there was a difference in their total distance moved and also in their speed. And then, but for the time that they spent in the different zones, in the corner zones and the middle zone, there was no significant difference between them. So we then next move on to the T-maze, which is used to test for working memory. We found out that also that there's a difference in the speed between the 2 mice. There's a significant difference. But for the time spent in the novel arm, because how we train the mice, we'll first train the mice with the familiar arm, and then a normal mice will spend more time in the familiar arm. But the transgenic mice or a disease model mice will spend less time in the novel arm as compared to the wild type mice. So when we now test for the working memory, the wild type mice will spend more time in the novel arm, but the disease model will spend lesser time in the novel arm. So when we conducted the experiment, we found out that the wild type mice spend more time in the novel arm as compared to the transgenic mice. So my plan now also for the distance as well and the speed as well, there's a significant difference in the 2. So my plan is to increase the sample size and then to see the significant difference, but it shows the trend. We can see the trend between the 2 groups. So next, we then move to the Morris water maze, which is used to test for the spatial memory. So we trained the mice first with the water maze according to the protocol, and they all learned throughout the time of the training. So for the probe phase, we will test them for the probe phase after the training. And in the probe phase, we discovered that there is no difference in the speed. And then the path length, it showed a trend for the transgenic mice to the platform. This is the distance to the platform. And then also the latency to the platform zone as well. The transgenic mice also showed a higher time as well compared to the wild type. And then the time spent in the platform zone, which means that the wild type mice, they actually remember where the platform is, and that's why they spend more time in the platform zone compared to the transgenic mice. So the frequency to the platform zone as well, we also found that there's a difference between the 2 mice compared to the transgenic, and then the time spent in the target zone. This means that the transgenic mice spend more time in the target zone, but did not locate the platform as soon as possible. So hence, this is the trend that we see here for the probe phase. So the reversal phase, what we normally do, is that we will change the position of the platform to the direct opposite side where the platform was initially located. So because of the shortness of time, that's why I didn't show these slides here. But for the reversal phase, we found out that when we did the reversal phase as well, there was no difference in their speed, but there's also the distance that they totally moved as well, no difference. But the path length showed a trend to the platform. That is the path length of the platform. And then for the latency to the platform as well, there is a significant difference between the transgenic mice and the wild type mice. So which means that the transgenic mice did not locate the platform as soon as possible. And that is what we expect. Then the latency of the platform as well, we can see the trend is higher in the transgenic mice compared to the wild type. And then the time spent in the target zone as well, there is no difference at this level. So my future work is that I want to increase the sample size first, and then I will then treat with the Dendrobium catenatum water extract and then examine its effect on the behavioral performance of the mice. So this is my work so far. Thank you.
Thank you. So there must be issue with the blood-brain barrier here, right? Not in general. So when you use some of the components of the water extract might not be able to get through the blood-brain barrier, and but if you see the effect, meaning that some ingredients, compound, do go through blood-brain barrier and get to the brain, right?
Yes.
So, but is that a concern in general? Again, I do not study neural medicine, but in general, how do people study this? And when they use first, as I said to the previous speaker, I think in vivo experiment is important, but now you're doing that. You're going to do that, and you're also going to do in vitro. But how do you deal with the issue that some compounds might not go through the blood-brain barrier to reach brain? But on the other hand, you say that you may not worry. You say that you don't. There's no. You're not going to solve that issue. Therefore, as long as there's an effect, then you want to focus on those, and eventually, hopefully, you get the active compound, right? And so that's one approach. But is that how you're thinking? Or?
Yes, Professor. Yeah, we are thinking that we will first treat the mice with the water extract and then observe its performance. If the performance improves, then we can speculate that it crossed the blood-brain barrier. Mm-hmm. And based on some of the studies that has been done previously, the dose that was administered to rats now, but not mice, showed that it improved the learning, and then it enhanced memory. So we also speculate that, if it can improve the behavioral performance, we can speculate that it crossed the blood-brain barrier. Then we can identify the specific compound that is involved in this process.
Okay, thank you.
Yes.
Can I ask one question? Why do you use the female mice? Because the female mice have the estrous cycle. So do you have think about the I just wonder why you use the female mice. This one is a preliminary study, so you use the female mice first?
From previous studies, we already know that the female gender, the AD, even in human, the AD progression is much more severe than in males. So even in the female mice as well, the phenotypes, we see them more in the female mice as compared to the male.
So now we will move on to the next section. That will be the discussion. But do the Professor Liu have any comment for each presenter? 那个刘院士有没有什么要再对我们每一位报告者的部分再提？
I think I made a comment on most of them. So with the exception of the first one. So the question I have is, well, not really question, the comment is, I'm still not clear why Zen humor, whatever it is, was selected. But I know that it has some in vivo effect. Therefore, I guess the student or professor are interested in knowing the mechanism. But my suggestion would be not look at the lipase only, but look at the more just like my comment to another student, that is these days take advantage of a high throughput approach. Yeah, do not look at the one thing at a time, but look at the overall effect, and you might find affect many different things. Then you can come up with a bigger picture. You can come up with a bigger picture. Yeah. Does it make sense? 是不是清楚？我想第一个 speaker 好像是 English speaking, so yeah.
其实人差群魔它在小鼠当中已经被证实说它就是具有降胆固醇的功效，然后它也有在很多细胞的 system 当中可以就是抑制像是 IDOL， 然后在肠道细胞当中也有抑制 NPC1L1 的功效。但我们就是想要进一步的验证说，那人差群魔它是不是也可以有降脂肪肝的功效，就是撇除了胆固醇这一块。
对的，那个就是那听起来会是比较 confirmatory， 就是知道的已经多了，就是相当多。那所以我还是建议还是一样，就是看不是问它会不会降低这个，而是去看更多的就是这个 consequence。
好，谢谢教授。
对，对，这个可能会，尤其是对已经知道的，知道就是文献越多的话，那这种只看单一的，可能就会比较impact 会就比较不是那么大。那如果说是趁这个时候去看比较多一点的，这它的影响。对，那现在的相当多的 approach 可以做。不管是这个你去看什么 gene based effect 什么 protein 啊，这些都可以做到的。那这个当然是会比较 challenge， 但是我觉得我自己也是鼓励自己的实验室都是尽量这样做。但是现在有不少 expert 就是以做这个为主，所以如果说是这方面如果中研院跟实际的合作有兴趣要做的话，我们这边我都可以介绍一下在生医所。做这个还做甚至做 single cell 的 study 啦，这些 RNA-seq 啦，就是一看可以看蛮多的这个。对，这个10年前我大概不会这样讲，现在的话这个 error 已经到了，所以就变成说是不用这种 approach 的话，可能就会比较disadvantage 这种想法。对，
好，谢谢教授。
所以想一下，不是那么难，因为不需要你自己做，就是machine做。对。对，只是说是就是想法可能可以不一样，对，那所以就是利用这个机会，所以有这些tool available。 然后如果说是要看 cell surface 的话，现在也是一下看个好多 color 的这种想法。对，像site of 的我一次看30几个。那另外我在参观这个林口长庚的时候，他用一个叫做什么一下忘记了，忘记那个名字，也是一个就是还有flow cytometry 的一个仪器。也是一下看30几个 color， 那也许不需要用到，我只是建议这种比较 high through， 看 multiple 的这个 effect， 不看只看一个，那这样也可以对机制会有比较容易去了解。对，
好，谢谢教授。
OK， 好。那另外刚才松鹤老师有 post 他的 condition knockout 事实上是 K mate with K14， 因此是 condition specific。那是说是以后会呢，还是就是已经是这种 approach？ 陈老师还在吗？
那个院士他好像刚下线。
OK， 是没关系，我不太确定。那学生回答吗？
学生还在。
不是问题是 is it so your professor mentioned that your mice has been crossed to K14, therefore it's excuse me, skin specific knockout, condition knockout, occasions are specific. Is it in the future plan or is it actually you're using those mice already?
Yes, Professor. We already proceeding the second period of mice.
Oh, so you're working on that. You don't have that yet.
Yeah, already prepared.
Okay, so that will be a very good approach. Yeah.
Thank you, Professor.
But the difference in the body weight is really concerning.
Yeah, that preliminary results show.
Specific condition knockout, you don't have that problem. Otherwise, it will be very hard to study.
Okay, thank you, Professor.
So the topical drug used to induce papilloma can induce squamous cell carcinoma, also melanoma, right?
Yeah, but the common case is the squamous skin cancer induced by the UV and chemicals. So we're focusing that squamous skin cancer. Also, squamous skin cancer is the 2020 years is the really common the cancer. Yes. They also located that only the epidermis area. It is a very disadvantage of our work. We cannot, if we checking the dermis area, it is a lot of work in our future. But squamous skin cancer is only located in the epidermis area, so it will be better we focusing that squamous skin cancer.
Yeah, my question is whether the model, I know I should check. I used to know that also induced melanoma in the model or not.
Yes, but it's not too much.
Oh, not okay.
Yeah, not too much. It's common is the squamous skin cancer. Common cancer is the squamous skin cancer.
Okay. Is it that TCP expressed in melanocytes?
Yes, but the not too much works in there, and still the melanoma and basal cell carcinoma and the squamous skin cancer, these three kind of the cancers, very unclear the rule of the TCTP.
See. Okay.
So we wanna checking that what is the mechanism of the TCTP in the skin cancer. Maybe we can check that. Next in our work will be in melanoma.
Yeah, okay, good. Thank you.
Okay, thank you, Professor.
好，那院士还没有要对其他的这个 presenter
没有 specifically 但是我想我们用中文讨论，因为难得徐立鹏老师在。我是说不是我中草药不是我的领域，但是我的对这个 in vitro study 是有 concern， 尤其因为我就是也是我在我的演讲里面一再强调，做 galectin 的就是如果是外加 galectin 到细胞，它造成的影响，我不太确定它是它的 in vivo 的真正的 physiological activity。那这个我也跟那个肖佩温老师谈过，这个很难 convey， 很难 convey。那为什么是这样子？因为它是一个比较 non-specific 的lectin， 然后所以可以引很多 molecule， 然后造成 response。那这种不见得会在 in vivo 发生，因为一定要在 right place right time。那所以这么多的文献在 in vivo 到底做什么的？应该是会有功能或重要的功能。但是到现在为止 ，hundreds of paper 没办法探讨到这一点，那需要某些 breakthrough， 在方法上面 breakthrough 才能把这个 galectin 在 extracellular 里到底做什么的，在人体里面、动物体里面，对，那也就如果我们 focus 在就是这应该不只是 lectin， 但是就是应该其他的 molecule 也都可能是这种 effect， 就是如果在人体里面的因为这些都是我跟你讲两个，一个是它是 compound formulation， 就是有很多，有些是可能是promoting 什么，有些是真正的君臣佐使的 concept， 所以都有，然后造成的。那在 in vitro 看到的跟 in vivo 的不见得是一样，那边的 cell type 里边如果是看某种 cell type 的 proliferation。但是在真正到动物体的时候，这种不可能就不是会发生的。那如果是提到这个 lectin， 那尤其是我对 plant lectin 真的很有兴趣，我觉得应该多做。它的功能真的不清楚。那如果说这个里面的 extract 里面有 lectin 的话，那它造成的影响，就是会就像我做 galectin 一样，这个影响可能在 in vivo 的时候就可能不会发生。对，这个所以因为这个我对这个的。特别想了很多，所以就是说是 in vitro 的实验就不太确定，对，这个是当然到最后希望能够 purify， 就是单一的 compound， 但这个在中草药这一方面是不是这样子？我不太确定，这不是 final 的 goal， 还是说 final 就是有一个比较纯化的。是一个 compound 的 formulation，complex formulation 应该是 complex formulation， 就只要最重要是有效果，而且是 standardization， 它是一直是从哪个来源来都是有效的，然后里面的 ingredient 也都清楚，然后它有效果，对，我是有这种想法，我也是跟有一些一些老师们谈过，包括郑永琪院士，包括 Edison Liu， 他是 Jackson 那边的，
是，
所以这个我觉得是不是值得讨论一下？就是做 in vivo in vitro， 要是我可能全部做 in vivo， 就是那样 purify， 就是到 in vivo 看有没有 activity， 在 in vitro 也许是某种supplementary， 但是那边可能会有刚才提到这种 concern， 对。
谢谢刘老师的 comment 哈，因为 basically 因为植物真的是一个很 complex 的 chemical reactor， 就是说植物里面的，因为我们都 focus 在 small compound 嘛哈，然后刘老师讲的是 lectin 是比较是 big molecular， 那我们现在就是在传统中药的或者是草药研究药用植都比较是 reductionist 的一个 science 的一个 practice， 因为on one hand, we want to see the in vivo effect. On the other hand, we want to know what are the active ingredients to cause the effect, which can inhibit, for example, cancer growth or metastasis. So we got to have a very quick and way to isolate or identify the most enriched fractions and also the active component. So for cancer cell, of course, if we can inhibit the cancer cell proliferation, that's an indication, but it's very preliminary, as you say. So 就是说方法学上面，我们势必要有一个方法能让我们去无存精。可是去无存精的过程中，我们就要 relate 到我们真的有效性是什么，所以不得不也会要有一个方法。当然， preparation is only preparation， 像 MTD 就只是测一些 dehydrogenase later dehydrogenase activity， 也不能代表go anywhere。可是确实是一个很好的方式来导引我们去找到我们想要找到的东西。那现在刚刘老师讲的 lectin 你已经有设定一个物在那里，而且是蛮大的物那它的 mechanism 跟很多的小分子其实不太一样的所以就是说，那我们是不知道我们是什么样的小分子有功效。这是一个。
所以你的答案事实上是有，我想我已经 provided answer， 就是你是会 focus 在 small molecule。
对，
一开始就是你的 extract 也是会是 small molecule extract 这样子。
Yes。So we use the way to enrich the small molecule as a fraction。
对，那个的话就是比较不会有，当然也会说是一些 effect 可能是几个 mixture 才造成的。
是，对。
那真的 concern 就不一样了。所以如果是在 focus 在 small molecule 的话，也是刚才最后一个 speaker EBIO 也是一样。我想就是他如果是focus 在 small molecule 应该是就比较没有我刚才这个 concern 了。对，
是。那如果我们也是想要看 lectin 这一类比较大的，我们就会 modify 我们最 initial 的 extraction protocol。它的萃取方法的 mindset 就会不太一样。
那有这个需要性嘛？就是会去找这个 larger molecule， 所以就是不是 focus 在 small molecule。
是，那
有这个必要性，还是说是不一定？如果说找 small compound 就够的话，也就没有必要找这个复杂。
其实找 small compound 就已经是一个 big effort
是，
而且大分子因为我本身不是那种 glyco 那个大分子的那个expert， 糖的那个 polymer 那些对我来说是比较复杂，所以我在实验室开办20几年前，我就 set on the small molecule as our target。OK。对，比较就没有去注重大分子。
如果说几个同学都是做 small molecule， 那我的这些刚才的 concern 就没有。但是也许并不完全。那 water extract 呢 ？water extract 是 small molecule 还是不见得是
？water extract 就两个都
有，所以那边就要小心一点就是了。
对，所以要
最后一个同学他是那个就是两个issue是不是 cross blood-brain barrier， 一个就是他 in vitro 的 assay 的话，包括比较 protein 啦这些 lectin 也都包括了，那就是那影响就可能就会有我的 concern 在里面。
对，所以他如果 further 的 fractionation， 他就要想好，他要 enrich 大分子还是小分子就要考虑。
OK， 谢谢，我在 learn the lesson。
谢谢。
OK。
好，我们今天的整个talk 就会到这边为止。不过在结束之前，我们还是要谢谢今天刘院士给我们的指导。